Okay guys, we've been learning about DNA, and today we're going to make our own little individual model of a DNA strand. What we've learned is there are 4 types of protein on a DNA strand. There's adenine, thymine, guanine, and cytosine. And today we're going to use beads to represent those 4 proteins. Cytosine and guanine are always together, and we're going to use the red beads for the cytosine and the green beads for the guanine. We're going to use this light blue beads for the thymine, and that is always paired with the adenine, and we're going to use the dark blue beads for those 2 proteins. And then on each side of the DNA ladder we have dextrose, which is actually deoxyribose, which is a form of sugar, so we're going to use clear beads for that, and then we've got some phosphates, so I've got white beads for that. Okay, first thing we're going to do on your tables, you should have a little dish of each type of bead, so I'm going to get that set up really quickly. The other thing you're going to need for today is 2 black chenille stems. We're going to cut one in half, and we're going to leave the other one its full length. Oh, and there is a mess up, so we'll get that out of there. Okay, so we've got our 4 proteins, and they're done in their pairs. And quickly, I need to get out some phosphates and some of our clear ones to represent our sugar. Okay, first thing we need to do is take one of these chenille stems, and we're going to cut it in half. Okay, it's got a little piece of metal in the middle, but it's pretty easy to just go ahead and cut it in half. Okay, those are the 2 sides of our ladder, and this one that we're keeping together in one piece is going to be the one that we build our DNA with. We're going to take the 2 ends, and we're going to hold the end of the short piece and the end of a long piece together, and we're going to use our scissors to make a little loop. That's so the beads will stop. And we need to do the same thing on the other chenille stem. Make a loop so that we have somewhere for our beads to stop. First thing then is we're going to put a sugar and a phosphate on the single stem and set that down. On this side, you've got the 2 together. You can do the exact same thing. We're going to start out with a sugar, and we're going to put a phosphate, but when we get down to this part, we need to make sure both stems go inside. And it's easy with the clear one, but it's not quite as easy with the smaller white one. But there you go. We've got our sugar and our phosphate on both. Okay. Next thing you're going to do is bend it. Bend the long piece, just the long piece over. And you're going to make a DNA protein pair. It doesn't matter which one you make. This is your DNA, so you can make it as individual as you are. And I've done, which one have I done? I've done the guanine and the cytosine for my first pair. Okay? So you've got that going sideways. We need to put another sugar and another phosphate on this side of the ladder. Push it all the way down. Now, take this long piece and push it up. Now we're going to bring our other piece over and do like we did over here. We're going to put a sugar, and we're going to make sure that it goes over both stems, and then we're going to put a phosphate, and we're going to make sure that that one goes over both stems. And now they're completely connected. And that's basically how you make a DNA ladder. We're just going to keep going on the process. I'm left-handed, so I'm kind of doing everything to the left, but you guys do it however it's comfortable for you. But here I am again, and I'm going to bend it sideways, and this time I think I'm going to do one of the other pairs. So I'll slide those 2 on. This pair is going to be adenine and thymine. So I'll slide that over, and there they are in the base. Put another sugar and put a phosphate on the single side. And remember, bend it up and hold those two together, because on this side we're going to put the sugar and the phosphate and make sure that both go in. Okay? And now we've got one of each of our DNA strand. However you want to do the rest of your DNA is up to you. I have the green and the red here. The next strand could be red over here, green over here. Or I could do another strand of adenine and thymine. It doesn't matter. It's your DNA. You do what you like doing. Just keep going with the two proteins. Remember to always put your sugar and your phosphate. Push this one up. Put your sugar and your phosphate and keep going. By the time you're finished, you're going to have, maybe 8 to 10 pieces or rungs on your ladder. Okay, I'm working quickly now because I want to show you how to finish it off. Always go back and put your sugar and your phosphate, your sugar and your phosphate, and always making sure that you get both chenille stems on that side. And it's getting a lot more sturdy, and it's a lot easier to work with now. I think I'm going to do 2 of these in a row. Okay, bend it up. Sugar and a phosphate on this side. It actually stuck in my thumb. Be careful of that that little piece of metal in there. Don't let it stick you and hurt, but it literally stuck to my finger. Kind of weird. Okay, go back to doing a cytosine and a guanine. Bend it up. Hopefully at this point this is you've got this and you're putting together your own beads as we speak. Okay, I'm just going to keep working. Bend it over. Sugar and a phosphate. We need to have room at the end so that we can make another little loop with the chenille stem so that we don't lose the beads on this end. Let's see, let's do another one of these, and let's do it with the, which one is this? Let's do this with the guanine first. Okay, my sugar and my phosphate. And you can see that once you've got the process down, it's really quite easy to make the DNA strand. I think I've got room to make one more. If not, I can always go back and take the last set of beads off. Okay, sugar and a phosphate over here. Bring this over. Oh, you know what I'm going to do? Right now, I'm going to go ahead and make that loop with my scissors and push it in there, and that way that side's finished and those beads won't come off while I'm trying to do this last side. Okay, one last sugar and phosphate on this side, and then remember you need to make the loop so that it includes both pieces of chenille stem so that you don't lose any of your beads. Okay, that's it. There you have your basic DNA ladder. But remember that the DNA strand is in the shape of a double helix. So what you need to do is take one side here, and give it a nice twist. And there you go. Now you've got your model in a double helix. Ta-da!